So once you generate a probe DNA that is labeled with something like a radioactive isotope, now you can do the process of southern blot hybridization. Okay, so this diagram showed you the entire process of southern blot hybridization. So you're starting from this. So it looks like what? It's an electrophoresis gel, agarose gel electrophoresis, right? So your DNA or RNA fragments could be separated out based on their size difference. So your DNA molecules will be located inside of a gel, and they'll be maybe sorted out on the gel, right? So then once that's done, the DNA, actual molecules, can be transferred from the gel to a different medium. In this case, maybe a membrane that's made of with nitrocellulose. Okay, so it's described there. Maybe the gel is located at the bottom, and then maybe nitrocellulose membrane on the top of it, and then stack of paper towels and filter papers. And then using a capillary reaction, they will move that buffer that fills up this bottom container. Okay, so the buffer will travel from the gel to the membrane, and from the membrane to the stack of paper towel. So while the buffer will travel, the DNA molecules will follow from the gel to the membrane and will try to go to the stack of paper towel, but membrane has very small pore, so the DNA will stuck on there. Okay, once that's done, you can peel up the nitrocellulose membrane, and then the DNA will be identified on the surface of the membrane. So that's why it's called southern blot hybridization. I explained what the hybridization means, right? It's the binding between the probe and matching DNA. Then what is the blotting? Blotting is the term describing this process. So blotting is the movement of DNA molecules from the gel to the membrane using this capillary reaction. That's what blotting is. Okay, so when you say southern blot hybridization, yeah, you move the DNA from the gel to the membrane through the blotting process, and then you're going to hybridize your probe DNA against some of this DNA. So another important thing here is when you use this buffer that will travel from the gel to the membrane and all the way through the paper towel stack, that buffer will have probably a basic solution like sodium hydroxide, and that solution is strong up so the DNA, if they were existing in the gel as double-stranded, that buffer will denature them, so the DNA will be separated into single strands. So not only it's a blotting, that denaturation can take place as well. Okay, by the time that's done, you're gonna soak this membrane with a neutralization buffer which has acid. Then the pH will be neutralized after that, and then once that's done, this membrane with the DNA transferred on the surface will be added into a maybe ceramic plastic bag, maybe sandwich bag like that, and then it will be added with the buffer that provides the neutral pH and some salt. Okay. And then these wiggly bits looking like all form, they are not the wiggly bit all form, they are the probe DNA that's radio labeled. These red single strand molecules are probe DNA. Okay, you add the probe DNA there, okay, and then seal that bag. So your probe DNA will be floating. So you put this back on kind of a shaker incubator and leave that there overnight. Then what's going to happen during the shaking? So your probe DNA may be able to find and then go hybridize on a DNA fragment that has a matching sequence. Okay, so the hybridization will take place slowly overnight. Okay, so once you come back about next day, you come back and open up the bag, and then take the membrane out, and then try to wash it up, because there may be some random unspecific binding of your probe on the membrane, or something else may happen. So you wash up those non-specific binding and leave the specific binding only. Okay, and then you do the detection of the location of your probe on the membrane by exposing this membrane to X-ray film. Okay, and the X-ray film is developed, and then if it gives you these dark lines, what that means is dark line shows where your probe is sitting. Okay, that probe sitting means yes, indeed, among your DNA fragments that are located in the gel, which after that they transfer on the membrane, they are matching DNA fragments to your probe DNA. So that was the identification. Okay, and when you do that, so why is that important? That's important that the existence of matching DNA between your probe and these DNA molecules. And not only that, if you could maybe duplicate your gel early at the beginning, if you have the same gel that is not used up for this process, actually you can go back using this extra film, put this extra film on top of your unused gel and match where this band is lining up. And actually you can get a gel slice cut up from your second gel, and then from that gel slice you can actually elute out your DNA and isolate that DNA and use it for your next step. Next step may be cloning or isolating that DNA, right? You can do that actually. Okay, so at the end of this process. So that is a sudden hybridization process. Okay, let's talk about it in detail. So let's say at the beginning you're using the entire genomic DNA, try to be spread out using the electrophoresis. Okay, so if entire genomic DNA is used directly for this hybridization, then genomic DNA isolated from the cell is first digested with one or more enzymes. So they will be linearized, and they'll be cut up into different size fragments, right? When you do that, you mostly need 4 base pair binding and cutting enzyme, because 4 base pair compared to maybe 6 or 7 base pair, they cut more often, so you can generate smaller fragments. Once you cut the genomic DNA, and those DNA will be separated out on a gel electrophoresis, right? So fragments are separated according to their size by electrophoresis on a gel agarose gel. Okay, so normally when you do that, so like this case, your cut up genomic DNA is added to the well, and then electrophoresis will move them and to opposite direction. But when they do that, they will be separated out on a gel. Okay, but since you have thousands and thousands of fragments, those fragments will show up as a smear instead of band pattern, distinctive band pattern. Okay, that's what I mean. Normally DNA appears as a smear on the gel rather than a distinct band, because you have so many fragments. Okay, and then DNA in the gel should be denatured before hybridization with the probe taking place, because right now your genomic DNA is existing as a double-stranded DNA. Okay, and then when they are cut with the enzyme, still they are double-stranded. When they are separated out, they are still double-stranded. So now you have double-stranded DNA, okay, located on this gel. Okay, but your probe is a single-strand DNA. Okay, if your probe to be hybridizing to the target DNA, your target DNA should be single-stranded as well. Okay, that's what we are talking about here. Okay, so you need to denature the gel, the DNA in the gel, into single-stranded form. Okay, the reason is why, because they should bind with your probe, which is single-stranded. Okay, then how are you going to denature them? By soaking your gel in a buffer that contains sodium hydroxide. Okay, DNA in the gel is transferred to a solid support, such as nylon or nitrocellulose. That's the blotting process we talked about. So it's this: once your gel is denatured and in this buffer, so you put your gel at the bottom of this container that contains the denaturation buffer. Okay, and then on top of the gel you place a solid support like nitrocellulose membrane, your filter membrane, or nylon filter membrane, doesn't matter, one of those two. Okay, and then lay a stack of paper towel. That's what we're talking about here. Okay, so that's for the blotting to take place. You're setting up the blotting, capillary blotting assembly. Okay, the DNA attached to the membrane is hybridized to so after the blotting, okay, so blotting is done successfully, so then the DNA molecule from the gel is moved to the nitrocellulose membrane. Then you need to assemble this blotting assembly and then peel up the membrane. Once that's done, then the DNA on the surface of the membrane will hybridize with your probe. So we're talking about that step here. That step is explained here. DNA attached to membrane is hybridized to a DNA probe that's labeled with some kind of signal generating molecule. In this case, maybe radioactive isotope. Okay. And then once that's done, the DNA band complementary to the probe is located by a detection method such as, the X-ray film. Okay, and we're talking about the last step. Okay, so once only the specific probe hybridization is identified, you put extra film on top of it. Now you can detect the radiation signal hitting the extra film when you develop this film. Okay. So then, by estimating the size and number of bands generated, it is possible to place the target DNA within the context of a restitution enzyme site. Okay, so what you're saying is, where your band is located, how large is it, and that gives you a clue where the DNA fragment matching on the sequence your probe is located. Okay, you can also go back to the location if you have a duplicated gel, and you can isolate those fragments out. That's what we're talking about there. Okay, does it make sense? So in the whole process, we used either nitrocellulose or nylon membrane. So those are the filters where your DNA fragments will travel from the gel, right? So which one is better? I mean, there are pros and cons in choosing one of these two. Nitrocellulose membrane, it binds DNA through hydrophobic interaction, and it creates less background. What does that mean? Less background. What that means is your probe can bind to the membrane itself without binding on the target DNA with the base matching. Okay, so nitrocellulose may bind with your probe DNA. So that's a background. So nitrocellulose tend not to do that. That's what it means. Okay. However, its binding capacity is low and become brittle during the baking process. So it doesn't bind a lot of DNA, and then when you have to dry out this filter after the blotting is done, because it's a hydrophobic interaction. For you to keep the DNA on the membrane surface, water molecules should not be there. So you have to bake it. Then once you bake it, this membrane becomes kind of brittle, so it can break down during your process. You probably don't want to do that. So there are pros and cons for the nitrocellulose membrane. How about nylon membrane? So nylon membrane binds nucleic acid irreversibly, so it can bind and you can strip them up, so it's more durable. Okay, and then transfer can be carried out. Instead of blotting, you can use electrophoretically, meaning if you have electrophorator, you can use the gel on top of the negative plate and then membrane on the stack of paper towel and the positive plate. You can do that same blotting using electric current. That's what it means. Okay, so nylon membrane is stronger, so it can go through that process, and then you have to, once that's done, you have to UV cross-link your DNA to the nylon membrane, because it's not the hydrophobic interaction that's going to hold the DNA on the nylon membrane. Instead, you have to UV cross-link. You have to expose nylon membrane to a strong UV light. Then DNA will bind on the nylon membrane. So that is an additional process if you decide to use nylon membrane. So like I said, the DNA in the gel should be denatured from the double-strand conformation to single-strand conformation. Okay, otherwise they're not going to bind with the probe DNA, which is single-stranded. Okay, so what kind of solution can you use? So the typically denatured solution contains high salt, 1.5 molar sodium chloride, and sodium hydroxide. Like I said, half molar sodium hydroxide. So strong alkaline condition can break up all those hydrogen bonds. Once that's broken, they'll stay a single-stranded conformation. So once that's done, your gel can be neutralized. Otherwise, they're not going to bind with your probe. So you have to neutralize it by soaking your gel and maybe membrane in a neutralization buffer that contains tris and salt alarmo salt. Okay, so that's a denaturation step. Understand why you need to denature the DNA. And then transfer. So the gel itself is, I guess, denatured and then neutralized. Once that's done, you're going to transfer the DNA from the gel to the membrane. So the capillary reaction is what it's going to carry the DNA from the gel to the membrane. So as you can see, so your gel can be seen at the bottom. It can be a podium, I guess, and your gel, and then your membrane, and then a stack of paper towel, including some layers of a filter membrane. So if the buffer only touches your gel, and then maybe some filter papers, and then buffer will carry This is not the membrane, I'm sorry, this is the podium. Okay, so the buffer will, travel through this membrane, maybe some kind of paper, and through the